Hey everyone, this is Vaibhav Chandila from ESIC Government Medical College, and today we'll cover the disease African sleeping sickness, which is caused by the organism Trypanosoma brucei complex. Okay, so we'll discuss this organism under the headings of introduction, pathogenesis, and lab diagnosis. So let's get started. So first of all, the Trypanosoma brucei complex responsible for African sleeping sickness. It actually includes Trypanosoma brucei gambiens, rhodesians, and brucei. Okay, these are just three names. If you want, you can remember it, but just remember brucei. And these are the Trypanosoma brucei complex, which are causative organisms. Now coming to the life cycle is a little tricky here, but there is a pattern which we'll recognize soon. So first of all, there is a bite of fly. After this bite of the fly, we get something called the metacyclic trypomastigotes in skin. Metacyclic trypomastigotes. They convert into the long slender trypomastigotes, okay, which divide by binary fission. Then they form short stubby trypomastigotes, which are non-dividing, which invade the bloodstream and go into various organs, and at the same time are also ingested by the fly again, and inside the fly they convert into long slender trypomastigotes again, then epimastigotes, and metacyclic trypomastigotes back again bite the human. So it's a little confusing, but the main thing you can see here is we're just rotating among the metacyclic, then long slender, then short stubby, and then reverse. Long slender, metacyclic. It's just like a cycle. Just to get an epimastigote form in the fly, which we were also getting in Chagas disease, if you remember. The epimastigote form was only in the vector, in the redovid bug. Similarly, in fly, we get the epimastigote form. Otherwise, it's just metacyclic, long slender, short stubby, again long slender, again metacyclic. That's it. Easy life cycle. Antigenic variation. So, our Trypanosoma brucei complex has antigenic variation. Basically, they have a variable surface glycoprotein VSG, which changes its structure from time to time over evolutions. And because of this is the key mechanism which helps it evade host immunity. It's always changing VSG. It shows antigenic variation. Next, coming to the pathogenesis, this organism infects in two stages. Stage 1, the hemolymphatic stage, and stage 2, the CNS invasion. Okay, so the first stage, where the hemolymphatic stage, here basically we see formation of a trypomastigote chancre, which is basically a self-limiting lesion, and it appears after 1 week of infection. Okay, it's a self-limiting lesion which appears after 1 week, the trypomastigote chancre. Next, you have the asymptomatic stage, which lasts for months, and then the systemic febrile illness, which again occurs months to years later after this phase, and it is characterized by fever and night sweats and posterior cervical lymphadenopathy, which is called winter bottom sign. Okay, so the systemic febrile illness has months to years later it occurs, and it shows fever, night sweats, and post-cervical lymphadenopathy called winter bottom sign. So this is the stage 1 hemolymphatic stage. Okay, we are affecting the lymph nodes also, and we are going in the blood also. We're causing fever, and we can see these trypomastigote chancres. Okay, then the next stage 2, CNS invasion. Now this stage is a more serious stage, where CNS invasion, if occurs, can show chronic meningoencephalitis. Okay, chronic meningoencephalitis, and at the same time we see nighttime insomnia while daytime sleepiness. Relatable. Nighttime insomnia and daytime sleepiness. This is the stage 2 CNS invasion. Okay, so these were the two stages. Next, we come to the lab diagnosis, final stage. Here, the specimen we take for diagnosis is the CSF, urine, blood, chancre fluid. Basic stuff. CSF, urine, blood, and the trypomastigote chancre fluid. Next, for blood examination, under thick, thin smear and wet mount, just like Chagas disease, we see the trypomastigotes under the wet mount and the thick, thin smear. Then, there is something called the concentration method for low parasite count, which we do using microhematocrit centrifugation. Something, if you want to remember, you can remember, nothing important. Microhematocrit centrifugation for low parasite count. Detecting low parasite count. Then CSF examination. Now this is important because the CSF examination will tell if CNS invasion has occurred or not. Now what are the criteria for CNS invasion? First of all, there will be detection of trypomastocytes, or there should be WBC more than 20 cells per microliter plus parasites detected in blood and lymph node. Okay, so either there should be detection of trypomastocytes or WBC more than 20 cells per microliter and any form of the parasite detected in blood and lymph node. Then we can say that CNS invasion has occurred. Next, you can take a lymph node aspiration for examination. Then antigen detection will be by ELISA, and antibody detection. Now, which antibody will you detect? So you will detect the antibody to the variable surface glycoprotein earlier, which was showing the antigenic variation, right? The antibody to that will be detected using card agglutination test for trypomastocytes, or CATT, card agglutination test for trypomastocytes, and ELISA, semi-quantitative ELISA. Okay, this will detect the antibody. Next, the molecular methods by PCR and FISH, fluorescent in situ hybridization, and we take a culture, we inoculate it on in mice, and something called KIVI, which is kit for in vitro isolation. So a lot of terms here in the lab diagnosis, but what we can remember is in blood examination, again wet mount, thick thin smear, trypomastocyte form is detected. Other than that, in CSF examination, we confirm CNS invasion by detection of trypomastocytes or a WBC count more than 20 cells per microliter and parasite detection in blood lymph nodes. Apart from that, we can detect antibodies against the variable surface glycoprotein antigen using card agglutination test for trypomastocytes, CATT, and then the usual antigen molecular, and we do a culture in the KIVI, kit for in vitro isolation. That's it for lab diagnosis. So this was African sleeping sickness caused by Trypanosoma brucei complex. It shows antigen variation. The life cycle involves metacyclic trypomastocytes, long slender, short stubby trypomastocytes, in the fly just an epimastocyte, and then back to long slender and metacyclic. Then pathogenesis, we saw two stages, the hemolymphatic stage and the CNS invasion stage. Hemolymphatic stage showed trypomastocyte chancre, asymptomatic stage, and then systemic febrile illness where we saw fever, night sweats, and post-cervical lymphadenopathy, winter bottom sign. CNS invasion, we saw chronic meningoencephalitis and daytime sleepiness and nighttime insomnia. Finally, we discussed about the lab diagnosis, how you detect the organism in various cultures. Okay, so this was the organism. Hope you liked the video. Thank you.